Click the bell icon and subscribe to our channel so that you will not miss any classes.
Hello students, welcome to class. In the previous class, we have discussed in detail how the restriction endonucleases exactly cut the DNA and creates a fragments of DNA, and those fragments of DNA can be used in constructing a recombinant DNA, further a recombinant cell. And now I am going to give a list of few commonly used restriction endonucleases which can create the blunt ends and the sticky ends. So let us take up them now. The first one is HAE3 and it is isolated from the bacteria Haemophilus aegypticus, and 3 refers to, as we discussed from the nomenclature of restriction endonucleases, it is a strain. And this is the restriction site for this enzyme. That is, it recognizes 4 nucleotide long DNA sequence, that is GGCC, which is a palindrome, and it will cut the fragment at between between G and C, and same here also between G and C. So you should remember, in both of the strands the restriction endonucleases will cut the DNA between the same nucleotides, not of different. So here, since it is cutting the DNA here exactly like this, there is no remains of small stretch of DNA. That means a small length of single strand is not remained. So it will create a blunt end. So where we have discussed that the creation of sticky end is favorable in constructing a recombinant DNA, but here it is creating a blunt end. So same here also, the next restriction endonuclease ALU1, which is extracted from the bacteria Arthrobracter luteus, and this also recognizes 4 nucleotide long palindromic sequence, which is AGCT, and it will cleave the DNA between G and C, same here also, hence it creates a blunt end. And if we go on observing the remaining restriction endonucleases like MSP1, which is extracted from Mozzarella species, here it recognizes this 4 nucleotide long DNA only, but it is cutting the DNA here and here between C and C, hence the cutting will be like this. So as a result, when the DNA fragments are separated, there is a creation of small stretch of single strand. So which means it is creating a sticky ends. So same here also VU1, it also cleaves the DNA and creates a blunt end like this. Same here for BAMH1, it is also creating the blunt end. SAL1 also, it is creating a blunt end. PST1, Providentia stewarti source organism, it also creating a sticky ends. So these are all creating the sticky ends. These two are creating the non-sticky or the blunt ends. So here the creation of a sticky ends will favor the action of DNA ligase. You may ask the question, why there is a need of only sticky ends, not the blunt ends? Because the majority of DNA ligase enzymes are capable of joining the sticky ends, not the blunt ends. There are very few. So hence biotechnologists prefer the sticky ends over non-sticky ends. That is only the reason. And apart from that, the mode of action will be the same. They are also creating the fragments of DNA. These are also creating the fragments of DNA. Only the difference is there is a creation of blunt end, but here there is a creation of sticky ends, which is favorable compared to this. So this is the list of some restriction endonucleases which are commonly being used. Apart from restriction endonucleases, there are other enzymes used in constructing a recombinant cell. Let us discuss them in detail now. Along with restriction endonucleases, one of the most important enzyme used in constructing a recombinant DNA is DNA ligase. DNA ligase is capable of joining any fragments of DNA. So, DNA ligases can join the two fragments of DNA, hence they are called as molecular stitchers or molecular glues. So, they are just acting as a glue to join the two fragments of DNA. It is working opposite to that of restriction endonucleases. Restriction endonucleases, what they are doing, they just cutting the DNA and creating the fragments. Hence, we can call them as molecular scissors. So, they are cutting the DNA, hence we can call them as a molecular scissors. Whereas, DNA ligases can join the separated DNA fragments, hence they can be called as molecular stitchers or molecular glues. In 1972, two scientists, Mertz and Davis, they isolated DNA ligase from Escherichia coli organism, bacteria, and they called it as E. coli DNA ligase, and they demonstrated that E. coli DNA ligase is capable of joining the two DNA fragments covalently. That means it is capable of creating a very strong bond between the two fragments of DNA. That means that much efficacy it is having. So, the efficacy of the DNA ligase is that it can join the two fragments of DNA covalently, so that the two DNA fragments are joined permanently, they cannot be separated easily. So, that indicates the term covalently. That means it is the strongest bond, right? So, DNA ligase, which is isolated from Escherichia coli, this was the first DNA ligase which was isolated. No doubt, the ligases will be present always, but for the first time isolation was done by these two scientists, and they demonstrated that ligases are capable of joining the DNA fragments covalently. And the drawback of this DNA ligase is, which is isolated from Escherichia coli, is it cannot join the non-sticky ends, that is the blunt ends. It can join only the sticky ends. But nowadays in in biotechnology, they make use of DNA ligase, which is isolated from T4 bacteriophage, which is a virus. So virus which can infect bacteria are called bacteriophages. One such bacteriophage is the T4 bacteriophage. So DNA ligase isolated from this is called T4 DNA ligase, which is widely used nowadays in biotechnological works because it can join both sticky ends and blunt ends. It could be blunt end or it could be sticky end. It can be joined by T4-DNA ligase very easily. So that's why this is a DNA ligase which is commonly being used in constructing a recombinant cell. So likewise, one more enzyme used in constructing a recombinant DNA is alkaline phosphatase. Alkaline phosphatase. You know that in DNA there is a phosphate group present at one end and hydroxyl group present on another end, and vice versa. OH group and the phosphate group, we refer them as 5' and 3', 5' and 3', 5' and 3', right? So here the presence of phosphate group in a fragment of DNA. Let us take here a fragment of DNA. Here it is a fragment having phosphate and here is hydroxyl group. And here is one more fragment which is opposite to this, phosphate and the OH. So they have created, it is created by, say consider it is created by treating the DNA with the restriction endonucleases. It has created a sticky ends. So if we leave this as such in a solution, if we leave this as such in a solution, then there is a possibility of self-ligation. Again they may join because there is presence of phosphate group here. So for that, this phosphate group should be removed in order to prevent the self-ligation. So to prevent self-ligation of DNA fragments, phosphate group should be removed. Phosphate group should be removed, and this removal of phosphate group can be done by alkaline phosphatase. Once the phosphate group is removed, only the OH group is present at 5' end. At the 5' OH group, there is an attachment of phosphate group, right? And that phosphate group, if it is removed, then there is a remain of OH group. So that for next action of DNA ligase, this will be facilitates the action of DNA ligase. But the important work done by the alkaline phosphatase is it will prevent the self-ligation of DNA fragments by the removal of phosphate group present at the 5' end. So this is the one more enzyme used in constructing a recombinant DNA. And one more enzyme used is DNA polymerase. DNA polymerase enzyme. So you have heard about DNA polymerase enzyme in molecular biology very well. That is, DNA polymerase is helpful in a synthesis of DNA. So there is an artificial synthesis of DNA fragments in the process called PCR, polymerase chain reaction. So that we are discussing further. So for that, for the synthesis of DNA, that means whatever the DNA we have isolated from a target organism, it will be available in very few amount. So to increase that amount, so there should be a synthesis of that DNA fragments in artificial condition or in vitro. So for that there is a requirement of DNA polymerase enzymes. So these are the list of enzymes which are used in constructing a recombinant cell. One is restriction endonucleases, which is very important, and DNA ligase, alkaline phosphatase, and DNA polymerase for the polymerization of DNA fragments or the synthesis of complementary copies of the DNA. So that's about the enzymes used in constructing a recombinant DNA. And in the next class, we shall discuss how the DNA fragments separated. So once the DNA is treated with the restriction enzyme, there is a creation of two fragments. Say for example, not only two fragments, there are many fragments created. So among them, we need only one fragment. So how it is possible to separate it? It is not visible to eye. So we cannot pluck it up and keep it away. So there is a special technique for separating these fragments from the solution. So from the solution, it is possible to separate the DNA fragments based on their lengths. So that is done by the technique called gel electrophoresis. So that we shall discuss in the next class.